So in this lecture, we're going to take a look at the support cells that are there to help hold neurons in place and to provide nutritional or other support to them. We're going to start by looking at the neuroglial cells of the central nervous system. And this is where there's a number of different neuroglial types here. The largest and most numerous of these neuroglia are the astrocytes. They make up almost half of the volume of the central nervous system tissues. So half the volume of the brain and the spinal cord is made up of these astrocytes. With a name like astrocyte, you would expect that they are star-shaped, and certainly they are. They have this kind of star-like shape with lots of projections or processes that reach out and connect neurons to the capillaries. And so they seal the gaps between the capillary cells. Capillaries in the brain are very leaky. They've got these spaces between them. And the astrocytes seal up that space, and they prevent fluids from leaking out of the blood vessels. And then they reach out with other processes, and they attach themselves to neurons, and they serve then as the conduit for bringing nutrients, ions, dissolved gases into the fluid around the neurons themselves. And they also can absorb and recycle neurotransmitters and other ions that are found there in the fluids of around the neurons and the astrocytes themselves. So they're responsible for nutrient and waste exchange between the blood and the neurons, and they regulate the constitution of the fluids in the extracellular fluid, if you recall. That's what ECF stands for, that extracellular fluid that surrounds the neurons. They can also act as guides to the neurons, stimulating neurons to form and maintain synapses, to make neural connections between neurons. And they contribute to something referred to as the blood-brain barrier. Because they seal up those gaps, the leaky spaces between the cells that make up the capillaries, then they prevent a lot of things that would get into other tissues from getting into the fluids around the brain. And that can be very protective for the neurons. So they're there as this barrier, so that the only thing that can cross that blood-brain barrier are things that are able to dissolve in lipids and then can move right across the cells, the endothelial cells of the capillary, and move right across those cell membranes, or anything that there is a protein, transport protein, that will move stuff from one side of the cells that make up the capillary walls to the other side, into the extracellular fluid that's around the neuron. So that blood-brain barrier is a really important function, protecting the integrity of the fluids around the brain. Astrocytes also will form scar tissue following an injury, so they help to heal up damaged tissue, say if there's a stroke or some other damage to the brain or to the spinal cord. And they help to hold neurons in place. And as I said already, I have it in two places, they can absorb and recycle neurotransmitters. Oligodendrocytes are a second kind of neuroglial cell. So astrocytes, they've got numerous projections or processes off of them. This prefix oligo means few. So few branches, dendro means branches, so they've got few branches coming off of them. So they're sort of like, they look a little bit like a small astrocyte, but they have many fewer projections or dendritic branches sticking off of them. And they reach out with these extensions. So here we have an oligodendrocyte. They reach out with their extensions, and when they encounter a neuron, and so there's a neuron running through here, there's a neuron running through here, the axon of a neuron. Those are all neurons. When they reach out, they act a little bit like, if you can imagine a slap bracelet, one of those bracelets that you just kind of slap it around your wrist and it wraps around. Well, they reach out and they wrap, so if this is the neuron, they wrap around and around and around and around and around and around that neuron, growing around it, and all of the cytoplasm is squeezed out to just the outermost layer. So what you wind up getting is multiple layers of nothing but cell membrane. So these layers here of cell membrane make this fatty coating that covers or surrounds the axon. So here you can see an axon, and then here you can see all the layers. And then all of the cytoplasm is squeezed out to the outermost layer. So here would be a nucleus. Well, I'm sorry, no nucleus, excuse me. The nucleus is back in the central portion of the oligodendrocyte. So they just reach out, and there's no layer of cytoplasm. So I was getting it confused there, I'm sorry, with a different way of forming this myelin sheath. So we get this really lipid-rich myelin sheath here that surrounds and insulates the axon electrically. And we'll see that it's all about electricity. How neurons work is all about electricity. So one oligodendrocyte can reach out and produce many of these segments of myelin sheath around an axon. So here would be one segment of myelin sheath. Here is another segment of myelin sheath. Here's another segment of myelin sheath. We'll talk more about the structure of myelin in just a minute. So oligodendrocytes, they reach out, they wrap, they form myelin sheaths. Microglia, these are tiny little small oval cells. They've got lots and lots of processes coming off of them, and they serve as macrophages. They encounter dead neural tissues or wastes or pathogenic material that somehow makes it into the fluids around or of the central nervous system, and they can engulf and digest these things that don't belong there. So microglia act to protect the neurons by removing materials that aren't supposed to be there. And when there is damage or inflammation in the brain or spinal cord, then you'll see a real proliferation of the microglia. There'll be lots of them there. And then the last of the neuroglia for the central nervous system are the ependymal cells. Ependymal cells look a lot like a epithelial cell. So here, this should look familiar to you from our tissue. So they're cuboidal cells. Sometimes they can take on a columnar shape. And they line the fluid-filled cavities of the brain. These are called the ventricles. We won't really get to talk much about ventricles until we get into the next chapter. But in the interior of the brain, there are several of these open spaces that contain fluid. And those open spaces are lined with ependymal cells. As well, there is a hollow space right down the center of the spinal cord, and it is also lined with ependymal cells. And in some areas, the ependymal cells are ciliated, not in all, but mostly where mostly up in the brain is where we see ciliated ependymal cells. So the ciliated cells cover these knots of capillaries. The knots of capillaries are the choroid plexuses. And so the ependymal cells cover up these capillary knots, and they pull from the blood components that then are used to produce cerebrospinal fluid. So the ependymal cells process the fluids of the blood there in those capillaries, and they push into the spaces they push fluid that is termed cerebrospinal fluid. And so it's at the choroid plexuses that we produce cerebrospinal fluid. They fill up the ventricles of the brain, and these ventricles are connected to each other. And so fluid flows from the ventricles and then down into this hollow central canal. And this is the lumen of the central canal of the spinal cord. And they form a single layer lining all of these hollow spaces. They're joined by gap junctions, and so there's a lot of exchange between the ependymal cells. Sorry, I keep moving forward accidentally. And so there's a lot of movement of fluids then from the blood into the cerebrospinal, or as cerebrospinal fluid, and then that cerebrospinal fluid flows down through the central canal of the spinal cord and then flows back up again along the exterior of the spinal cord and then around the exterior of the brain. And up at the top of the brain there's a region where we reabsorb that fluid into the blood. And so there's this constant flow of cerebrospinal fluid that's produced by the ependymal cells that are covering the choroid plexuses in the ventricles of the brain. Okay, so that's it for the glial cells that I want you to be aware of in the central nervous system. They are the astrocytes, the support cells forming the blood-brain barrier, the oligodendrocytes that form the myelin sheath around some of the axons in the central nervous system, the microglia that act like macrophages, and the ependymal cells that produce cerebrospinal fluid. Now, there are two neuroglial cell types that I want you to be aware of in the peripheral nervous system. The first are the Schwann cells. Schwann cells form the myelin sheath in the peripheral nervous system, and they work just like the oligodendrocytes do. They encounter the axon of a neuron, and then they grow around and around and around and around and around, making a very thick layer of fatty, phospholipid material that insulates a section of the axons of a neuron. These are the ones that each section here of the myelin sheath that we find in the peripheral nervous system is a single Schwann cell. And so the outermost layer here is where all the cytoplasm is. Here you can see the nucleus of the Schwann cell. And so there's lots of cytoplasm in this area that is in the outermost layer forming the myelin sheath. And here we see, this is the axon, here's the axon itself. And it turns out in this one that there's some fluid that's directly around the axon. But this is the myelin sheath in this image. Okay, so you can see all these things that I'm trying to show you, hopefully. Okay, so each Schwann cell in the peripheral nervous system forms one little segment of myelin sheath. And it also forms this neurolemma that surrounds the entire segment of myelin sheath. And these not only insulate, but they also, if the axon gets damaged, they remain in place and help to direct the growth of a new axon. We'll look more at that in a later lecture. So Schwann cells are found in the peripheral nervous system, as are satellite cells. Satellite cells are small cuboidal cells, and they surround the cell bodies that are found in ganglia of the peripheral nervous system. And they help to control the environment there around the cell body. They provide electrical insulation around the cell body, and they serve as a pathway for the exchange of gases and nutrients and wastes. So their function is a little bit similar to the astrocytes that are there in the central nervous system, of helping to control the environment around the ganglion. Okay, so let's talk specifically about the myelin sheath. The myelin sheath, again, is provided by the oligodendrocytes in the central nervous system, and the Schwann cells in the peripheral nervous system. So this myelin sheath is a whitish-colored, fatty, segmented covering that's found around the axons, especially in long axons or large diameter axons. It's because it is cell membrane, it's composed primarily of phospholipid, but there is a fair amount of protein found in myelin sheath as well. And its job then, so here's the axon, and here are the segments of myelin sheath, either provided by a Schwann cell or provided by an oligodendrocyte. And they surround, and remember it's multiple layers, so they surround, again, this slat bracelet kind of idea. So it prevents, now, we haven't talked yet about it, but an action potential is actually this movement of ions from one side of the membrane to the other, followed by, first it's a movement in, and then it's followed by a movement of ions out. And so that movement of ions creates a current, and it sets up this electrical differential called the membrane potential. It sets up this differential on either side of the membrane. And the function of the myelin sheath is to prevent accidentally, or one of the functions is to prevent accidental, like cross contact of two axons, so that an action potential that started on this axon doesn't stimulate accidentally an action potential on this axon. And so this myelin sheath helps to prevent that from happening. That's the electrical insulation function that we associate with the myelin sheath. Now, in between the segments of myelin, we have these unmyelinated portions that are referred to as the nodes of Ranvier. So Ranvier, French dude is my guess. I don't know that for sure, but I would bet dollars to donuts that Ranvier was a French scientist who was instrumental in explaining the function of these nodes. And because the myelin is here covering the axon, what we'll see eventually is that it increases the speed of the nerve impulse, because the only place that an action potential has to actually happen is here at the node of Ranvier. Then there's a big leap, and then action potential, big leap, action potential, and so on. So the myelin sheath really helps to speed impulse transmission, speed action potentials down the neuron's axon, sometimes by as much as 100 times. It's a pretty big difference in speed. Okay, so in the peripheral nervous system, remember it's the Schwann cells that produce the myelin sheath, and each Schwann cell produces, each Schwann cell produces a segment of myelin sheath. There's very little cytoplasm found in the layers of the myelin sheath. Just little bits of cytoplasm. It's primarily just phospholipid and protein. Most of the cytoplasm is found up here in the outermost layer, and that area is referred to as the neurolemma. Again, the nodes of Ranvier are the gaps, and the Schwann cells cover the internode areas, the region between the nodes. And if we have a collateral branching somewhere off of this axon, it's always going to branch at one of the nodes. So here we go. So it's right here at the node itself that the collaterals branch off. And finally, there is a thin layer of fibrous connective tissue that surrounds the entire, both the Schwann cell and the node of Ranvier, and that thin layer of fibrous connective tissue is the endoneurium. So neurolemma is the outermost layer with the cytoplasm containing layer of the Schwann cell. The endoneurium is external to the neurolemma, and it forms sort of a strengthening cover. So in the central nervous system, the myelin sheath is formed, as we talked about already, by processes coming off of the oligodendrocyte. And so here we see an oligodendrocyte that's been cut. This is the nucleus there. And here are processes. Here's a process that reaches out and does that slap bracelet thing of surrounding round and round and round. Here we can see the cut end of a segment of the myelin sheath. The nodes of Ranvier are also found here, but they are farther apart from each other than they are in the peripheral nervous system. Because all the cytoplasm, or the bulk of the cytoplasm, is found here in the main body of the oligodendrocyte, there's no neurolemma that surrounds the myelin sheath, as we saw in the peripheral nervous system. And there's also no endoneurium either. Okay, so there we have it. The neuroglial cells of the central nervous system, the astrocytes, astrocytes, oligodendrocytes, microglia, and the ependymal cells, the neuroglia of the peripheral nervous system, the Schwann cells and the satellite cells, and then a little bit about this myelin sheath. And I recognize we haven't really explained myelin sheath very well, but right now I just want you to understand the structure of them. We're going to see their function in some later lectures. Okay? So, any questions? Make contact.